Inside every single one of us, right now, there's this incredibly sophisticated security system working 24/7. Its whole job, stop cancer before it even gets started. Today, we're going to pull back the curtain on our body's own internal guards, the tumor suppressor genes. It's a pretty wild thought, isn't it? That our own cells are in a constant battle to prevent some of them from going rogue. So how does this invisible war actually work? What are the tools our body uses to keep us safe? Well, the heroes of our story are these right here, tumor suppressor genes. The absolute best way to think about them is like the brakes on your car. In a healthy cell, they keep everything moving at a safe speed. They can pause the whole operation to make repairs, or if things are just too broken, they can even trigger a self-destruct sequence. So here's our game plan for this explainer. First, we'll meet the most famous guardian of our DNA. Then we'll check out the gatekeepers that control all the growth signals. After that, we'll get to know the specialized DNA repair crew. And finally, we'll see what happens when this entire system starts to break down one piece at a time. All right, our first stop is the commander-in-chief of cell security. This is a protein so important, scientists literally call it the guardian of the genome. Its name is P53. And that nickname is not an exaggeration. Just look at this number. Almost half of all human cancers involve a mutation in this one single gene. Seriously, if you want to understand cancer, you have to understand P53. And this chart just shows you how common this problem is. I mean, look at some of these cancers: esophageal, ovarian, colorectal. A faulty P53 shows up in nearly half of all those tumors. It's a major recurring vulnerability in our cellular defenses. So what is its job exactly? Well, P53 is a crisis manager. When our DNA gets zapped by UV rays, or hit with chemicals, or just makes a random mistake, P53 is the first responder on the scene. Let's trace this out on the diagram here. Normally, see that protein at the top left called MDM2? Its job is to constantly get rid of P53, just to keep its levels low and in check. But when DNA gets damaged, a sensor protein called ATM springs into action. Follow that red arrow. ATM basically tackles MDM2, blocking it. This allows P53 to build up, and that's the signal for it to get to work. Now, at this critical moment, P53 has to make a huge decision, and it has two main options. On the left, it can turn on a gene called P21. Think of P21 as slamming on the brakes of the cell cycle. This gives a cell some breathing room to make repairs. But if the damage is just too bad, it goes with the option on the right. It activates a different gene, PUMA, which triggers what's called apoptosis, a clean, programmed cell death. It sacrifices one cell to save the rest of the body. But what happens when the guardian itself is broken? This is a really fascinating and dangerous weakness. See, the P53 protein has to work in a team of 4. And with something called a dominant negative mutation, just one bad protein in that squad of 4 can poison the well, sabotaging the entire complex and making it completely useless. It's the ultimate one bad apple spoils the bunch scenario. Okay, let's move on to our next line of defense. Because it's not just about crisis management. We also have gatekeepers. These are genes that control the day-to-day stop and go signals for cell division. Let's look at the APC gene, which is a huge deal in colon cancer. In the normal cell on the left, you can see APC is part of this destruction complex. Its whole mission is to destroy another protein called beta-catenin, and that keeps a really powerful growth pathway turned off. Now, look over at the cancer cell on the right. When APC is mutated, that whole destruction complex falls apart. Beta-catenin is free, it builds up, floods the nucleus, and gets that growth pathway stuck in the on position. Here's another gatekeeper, P10. It's basically the brakes on a pathway that tells cells to survive. So, a protein called PI3 kinase turns PIP2 into PIP3, which then activates a powerful pro-survival signal called ACT. P10's job is super simple, but absolutely vital. It just does the reverse. It yanks a phosphate off, turning PIP3 back into PIP2, shutting the whole signal down. So you can imagine, without P10, that survive signal is just screaming all the time. So we've got crisis managers and we've got gatekeepers. But what about when the DNA damage is really, really bad? For that, we need to call in the specialists, the DNA repair crew. When a chromosome suffers what's called a double-strand break, this is a catastrophic failure where both strands of the DNA ladder just snap. The cell calls in the heavy machinery. You can see at the top that our old friend, the sensor ATM, activates a protein you've probably heard of, BRCA1. BRCA1 then comes in and helps prep the broken ends of the DNA, getting them ready for the actual repair. And this part is just so elegant. Another famous protein, BRCA2, brings in a repair specialist called RAD51. What RAD51 does is incredible. It takes the broken strand and uses the other healthy copy of the chromosome as a perfect template. It's like having the original blueprint to rebuild a collapsed bridge, bolt for bolt. It ensures a perfect, high-fidelity fix. When BRCA1 or BRCA2 are broken, this amazing system fails. Okay, now we can finally see the full picture. Cancer isn't just one thing going wrong. It's a cascade of failures. It's what happens when multiple layers of this internal security system get breached over a long period of time. This diagram of colon cancer is the classic example. It doesn't just happen one day. First, a gatekeeper like APC fails. That causes a small benign growth. Then maybe an accelerator gene, Cas, gets stuck on. Then another suppressor, SMAD4, gets knocked out. And finally, the guardian itself, P53, fails. Each one of these hits allows the cells to get more and more abnormal, more and more aggressive. This table really just summarizes those key players we just saw. And notice how they all do different things, right? APC messes up growth signaling, SMAD4 messes up a different pathway, and losing P53 just unleashes total chaos and instability. The big idea here is that it takes failures in multiple, separate systems for a cell to become a full-blown cancer. So let's just recap our internal security system. We've got guardians like P53 that are the crisis managers. We have gatekeepers like APC that control the normal flow of traffic. And we have the repair crews like BRCA1 and 2 that fix the really catastrophic damage. The most important thing to remember is that cancer only wins when this entire layered defense is taken down by a series of hits over many years. And as this overview shows so well, DNA damage creates two big problems. You get oncogenes, which are like a stuck accelerator pedal, and you get inactivated tumor suppressors, our broken brakes. You really need both things to go wrong for a cell to completely lose control. And this all leads us to a final, really hopeful thought. For decades, our main strategy against cancer has been to poison it or blast it with radiation. But as we get better and better at mapping these incredible, intricate pathways, the real question becomes, what if the future isn't about destroying cancer, but repairing our defenses? Could we one day learn how to reactivate P53 or fix a broken BRCA gene, basically rebooting our body's own security system to win the fight for us? The potential is just enormous, and it all starts with understanding these elegant guardians that are already built right into our cells.